So in this video, I want to do the test review for specifically chapter 13 here. First three problems associated with chapter 13. And along the way, I'll expand on some of the topics here and there. Now remember, when we talk about especially our monosaccharides like fructose here, we can draw the Fisher projection or the Hayworth. Now we're wanting the Fisher projection here. Now I don't want to draw L-fructose just right away. Let me actually draw the one that's more common, and that's D-fructose. Now fructose is a ketohexose, so that means that you have a ketone group up top, which includes this, CH2 with the hydroxyl group, then the carbonyl, and then, that would be chiral carbons through here. So with Fisher projection, remember we just use the little intersecting lines. But there's the ketose feature right there. We can just highlight that. So the ketose portion is the fact that you have a carbonyl group with 2 carbons on either side. We do have 6 carbons, 3, 4, 5, 6. Now the pattern down here for D-fructose, remember, right? We're drawing D-fructose first. So all D sugars have this hydroxy group on the right-hand side, or I should say the hydroxy group coming off the lowest chiral carbon is on the right-hand side. And then to define whether it's fructose or some other monosaccharide, then it depends on the pattern of the other hydroxy groups. In this case, there's an alternating pattern as you march up the chain here. And then hydrogens fill in all the rest. There, fine. So there's our D-fructose. Now, to draw L-fructose, what you have to realize, and then a lot of times you see this, it'll be like this dotted line. It's supposed to represent a mirror. So L-fructose, yes, it definitely means that this hydroxy group is going to be on the left-hand side, but it means more than that. Okay, when you say L-fructose, it means that every chiral carbon has been switched. So what this looks like here for L-fructose, we still have that same pattern up top. I mean, in some ways, we still have that core Fisher projection. That's fine. Again, L, yes, lowest hydroxy group on the last chiral carbon, it's going to be on the left-hand side. But again, it means more than just this on the left-hand side. It also means that that position and that position is going to be switched. And then fill in the rest for hydrogens there. If I had only switched this hydroxy group to over here and then left these 2 hydroxy groups in that position, in their original position, I would have a L sugar, but I don't know which one it is. I'd have to look it up. There's a bunch of these, so I can't keep track of them all. But I know I would not have L-fructose. Again, when we talk about enantiomers, when we talk about mirror images, which is what we're talking about when we go from D to L, the orientation, the spatial arrangement around each of the chiral carbons are switched, okay? And then last little note here, when you have, and it doesn't matter which one, but whenever you have the Fisher projection and you're using the intersecting lines here, that tells you that you have a chiral carbon at those positions. Because remember what it takes to be a chiral carbon. It means that there are 4 unique things attached to that carbon. And I could pick any 3 of these. It doesn't matter. Let me just pick one. Right? That hydroxy group is unique with respect to the other group on the other side. These 2 are unique, not to that carbon, but you actually, remember when we talk about chirality, when we talk about chiral carbons, we're looking at the entire chunk of the molecule that's attached to the chiral carbon. So when I box in 1, 2, 3, 4 groups here, just by eyeballing it, this looks different than all the other 3. That looks different than the other 3, etc. And I could do the same game for both of those other chiral carbons, which I won't because it takes up too much time and room. All right, that's what's defining a chiral carbon for us. So in this example, in the textbook, in the class, we learned about 3 different disaccharides: maltose, lactose, and sucrose. This disaccharide is something else, and I'll expand on that once we sort of answer these questions here. So even though we're presented a new disaccharide, or for that matter, a trisaccharide or something larger, we can still use what we learned about the disaccharides in class to identify like glycosidic bonds, what monosaccharides do we have, is it a reducing sugar, etc. And that's what this question is trying to get at. So first off, let's just do the identify the glycosidic bond. So the glycosidic bond is this guy here. Okay, right, that's an ether linkage that's connecting one sugar to the other sugar. And what we have to do, well, one thing, we need to number these carbons. Now, I will say that if you Google this topic, you will find at times these these 6-atom ringed sugar molecules like flipped over, presented a little bit differently in some examples. That's not what's going to be presented to you on the test for this class. So it's always going to be this particular pattern, 1 to 6. So in terms of the glycosidic bond, well, one thing we see, we have the 2 numerical values, right? Any glycosidic bond, alpha, beta, 1, 2, beta, 1, 4, right? You have numerical values. So for first question here, you have numerical values. This is a 1, 2 connection. And then sometimes you have something more, okay? An alpha or a beta. Or in some cases, like for the case of sucrose, alpha, beta, 1, 2 glycosidic bond. You have 2 of those anomer labels. And what the anomer label here is referencing, it's not the anomer that you see for the overall disaccharide. We'll get to that in a minute. It's the anomer right here. Which direction is this hydroxy group pointing right just before the glycosidic bond form? Well, it's pointing up. And we learned that if it's pointing up, that's a beta label. So this is a beta 1,2 glycosidic bond. Now, for our examples, if you see this cyclic ring of 6 atoms, it's just going to be this number 1 position or this number 1 position that can undergo that alpha beta mutarotation kind of reaction. All the other ones are locked in place. So yes, the hydroxy group was originally pointing down at the number 2 position, but it's always pointing down. That's what happens. All of these are locked into place. It's just this position that can pop up and down, or I should say this is position that can go up and down, or undergo mutarotation in the monosaccharide form. So it's just this position that I need to indicate the relative orientation. Let's identify the monosaccharides within this sample. Now, there's only 3 monosaccharides we learn: D-glucose, D-galactose, and D-fructose. Well, fructose, if I was to draw the Hayward structure here, D-fructose was the one that had the 5-membered ring with the various, groups coming off of it. Neither of these are fructose. So it has to be either D-glucose or D-galactose. And the way we distinguish one from the other was the relative positions of the number 4 carbons. So if you have that pattern of top, bottom, if you have an alternating pattern, that is D-glucose. If I look at the pattern here, well, this 4 position is the only one that changes. It's on top here, it's on bottom there. All the other positions, number 3 position, hydroxy group's on top. Number 2 position, this hydroxy group is on bottom. This hydroxy group was originally on bottom in the monosaccharide form. So it's just this position that changes, and that's D-galactose for us. Now, D-galactose, D-glucose would be fine if we're talking about a Fischer projection, but we're talking about a Hayward structure here. So we also need to factor in which way is the hydroxy group at the number 1 position pointing. If it's pointing up, it's beta. This one's pointing up, it's also beta. If this was pointing down, then it would be an alpha D-galactose. So anytime we deal with anything looking like a Hayward structure, if that position at the 3 o'clock position, or number 1 carbon in the case for galactose and glucose, if it's open, then you can have this beta or alpha label. And then finally, is this a reducing sugar? So a reducing sugar, that means that it can undergo mutarotation. I'll define that a little bit more in a second. In the lab and in the classroom, what we saw was that the mutarotation is you get a positive Benedict's test. That was the reaction. It went from blue to red. What is needed to allow mutarotation to take place? Well, what's needed is that one of those positions, again, for galactose or glucose, a number 1 position has to be free. Okay? It can't be involved in a glycosidic bond. So the fact that this number 1 position, that means that it can open and close and it can undergo mutarotation. If it has that feature, then we call it a reducing sugar. So really what I did here is basically the explanation. The answer is yes, it is a reducing sugar. Now, if the linkage had been between this number one position and that number one position, where both of them were involved in the glycosidic bond, then that's the only time when we have a non-reducing sugar. Okay, so that's one change that would happen here. As a little side note, if that was the case, if the linkage was between these two, and I should say the glycosidic bond linkage was between these two, then you would also have like an additional, alpha or beta in front of the label here. Okay, we learned about 3 different condensation reactions for the formation of disaccharides. One for maltose, one for lactose, one for sucrose. For the cases for maltose and lactose, then they are reducing sugars, therefore there is an enantiomer tag that's in front of their names. So theoretically, you could have alpha maltose or beta maltose or alpha lactose or beta lactose. Remember, the alpha beta is just indicating what that freed up position is how it's going to be pointing. And you'll see it in this structure. Sucrose is a non-reducing sugar. There are no enantiomers of sucrose. Both of those positions are locked in place. It can't undergo mutarotation. So drawing the condensation reaction here, that's the reaction for the synthesis of the beta lactose. So beta lactose is a combination of galactose and glucose. Now the way it's presented to us in the textbook, you have galactose on the left-hand side, so I'm just going to go ahead and place it on the left-hand side on the reactant side. Actually, before drawing galactose, let me do the D-glucose. So this is the way I learned how to draw these sugar molecules. I always use glucose as a reference, and then I remembered certain changes about glucose that then derived galactose and then other sugars. Glucose. All of these simple Hayward structures always has the CH2OH just to the left-hand side of the oxygen that's in the ring. It's always pointing up on the top side of the structure. And then for D-glucose, there's an alternating pattern. Top, bottom, top, bottom. I'm going to pause on this just for a second, okay? I'm going to leave this structure as is. And then the only difference between glucose and galactose is at the number 4 position, which I addressed above. By the way, these two structures are referred to as epimers. Everything else is going to be the same, at least until you get around to the number 1 position. That may differ. Now, the glycosidic bond between these two molecules, let's just actually go ahead and write that out. So let's see, the top of glycosidic bond. It is a beta. It's a beta 1,4 glycosidic bond. Well, what does that mean? So if it's the 1 and the 4 position that are going to link together in that ether structure, the number 4 is locked into place. It never varies. For glucose, it's always pointing down. It's the number 1 position that can vary, that can undergo mutarotation. Well, what does it mean? Beta 1, it means that this structure was originally pointing up. So what's going to happen is a glycosidic bond is going to be seen where the covalent bond goes in the upper direction and then connects to the bottom part of this structure. And then usually what we see here is that then, like these two structures are shifted. This was shifted down, this was shifted up. Let's do that here. Let's focus on the glycosidic bond for right now. So this is going to be pointing up, going up to the number 4 position, like that, and just sort of ink in what we already know, just to match up over here. And actually, we'll just even put that in orange. Why not? That's the only part that changes. All these other positions are the same. Making sure to point this one up because it's galactose. And then, since it's a condensation reaction, that means water is kicked off in this reaction. We're almost done. The only thing I haven't accounted for is the beta language in front of the lactose. So I'm talking about now lactose here. Beta lactose. What does it mean? Beta means that for glucose or galactose, alpha beta, that's always in reference to what's happening at the number 1 position. Beta is pointing up. So if it was pointing up here, then I'm going to go back and ink this in. That's one difference when I paused on this earlier. I wanted to put this beta in context with the structure and then backtrack and say, what did that mean about the reactant? Now, this question is answered. Let me just fill in a little bit more. So this would be beta D galactose, and then this is beta D glucose. And it forms, again, beta lactose. One other thing, it's not in the problem, but, pretty typical for me. Right, the reverse reaction here would be hydrolysis. And there are a lot of hydrolysis reactions in the body. There's a lot of water in the body. Okay, so enzymes coming along using water to attack some kind of linkage here to then break it apart into simpler molecules, hydrolysis. So let's talk about some of the structural features for polysaccharides. That's what we have here. All of these are polysaccharides. So amylose and amylopectin are the two different polysaccharides that decompose starch. Right? That's the energy storage molecule in plants. Glycogen looks a lot like amylopectin. This is the energy storage in animals. And then cellulose is a structural molecule in plants. Let's talk a little bit more specifics about the structural characteristics. So actually, let me try to draw a couple little sketches. So when I talk about amylose, it's like a single helical structure versus amylopectin. It has, you can think of a sort of a central helical structure here, but it also has additional helical structures coming off at different positions. Okay, this is more highly branched. In starch, amylopectin makes up about 80% of starch. So if I want to talk about the distinguishing structural feature, well, what I would say is that, amylopectin is branched. And we can be a little bit more specific than just branched. These branching points here, they're actually composed of alpha 1,6 glycosidic bonds. Now the distinguishing features between glycogen and cellulose. Well, I said the glycogen looks a lot like amylopectin. So you have this kind of you can imagine a central pigtail, central helical structure. Right? So that's kind of representing what I drew here. Well, in glycogen, you have more of these branching patterns. Now, it doesn't answer the question here, okay? But again, I'm using this question as just sort of a open-ended question in a way, just to cover all my bases for the structural features for all of the polysaccharides. Now, more specifically for this question, glycogen versus cellulose. Well, cellulose, it's like linear structures that sort of stack on top of each other. So that's one thing. They look dramatically different. And so in glycogen, just like for amylopectin, we do have the alpha 1,6 branches, but we also have like alpha 1,4 glycosidic bonds that make up the bulk of the glycosidic bonds within the pigtail. The reason why I wrote it here is because that's true for amylose and amylopectin. So amylose, amylopectin, and glycogen all share these alpha 1,4 glycosidic bonds that make up the main helical structure. Now, getting back to my question, and the final point is that the difference between glycogen and cellulose here, cellulose, it has beta 1,4 glycosidic bond. Right? So it's just a change of alpha versus beta that produces a helical structure versus a linear structure for these polysaccharides. And the last thing I want to say, one thing that's common to all of them is that you have D-glucose as the monosaccharide. And that's all of chapter 13 in the test review here. And we'll have a second and third video for the other two chapters. Okay, thanks.